No, this one is fine. I'm easily technologically challenged, so if I stand here and the gears are visibly grinding and you see the smoke, just bear with me. It just means that I've been boggled by either the clicker or the keyboard. So thank you to the Tallgrass Prairie Center for the invitation. As Laura said, my name's Shelby Flint. I'm an assistant professor of biology at Southwest Minnesota State University, which is in Marshall, roughly 40 miles east of Brookings, South Dakota, and about 70 miles north of Worthington. So it really is the pretty far southwest corner. The work that I'm going to be talking about today is work that I did, however, as a graduate student or as a postdoc at the University of Minnesota with various collaborators, and I'll be talking about that information as well. And most of that was funded by Minnesota's Environment and Natural Resources Trust Fund, which as far as I know has been re-upped. So any of you who are interested, who are from Minnesota or interested in doing research in Minnesota, it's a really useful source of funds, especially with increasing uncertainty with regard to federal and state funds. And then the other thing I want to say is I realized belatedly after talking to Laura and sort of pitching my talk that I had bitten off a bit more than I could chew. And so the diversity aspect of this that I'm going to be talking about that I refer to in the title is not going to be the functional diversity that I talk about in the little blurb about the talk. I'm going to be taking a slightly different tack on that. So overall, what my work and the work of the folks I collaborate with has focused on is kind of these different questions that all of us, right, as researchers, as seed producers, as practitioners, want to try to get a better handle on, and hopefully a handle on in collaboration with each other, right? Ideally, researchers would be more responsive to what practitioners and producers need to know in terms of hard and fast data. And I promise you, we are working on it. Some of these figures are going to look very much as they are pitched at the scientific publication community, but we really are making an effort to have results and some actual applicable guidelines or suggestions is probably a better word coming out of some of this work. So what the work that I'm going to be talking about today, what it focuses on is how does distance and diversity in seed sourcing affect plant fitness, so survival and reproduction, we kind of consider those jointly, or performance, just in terms of establishment, in terms of vigor in the field, vegetation community, especially vegetation community diversity, and a couple of aspects of ecosystem function. I'm going to be focusing on biomass, so net productivity in regard to that. And here when I say distance, I'll pull this apart a little bit, as you'll see in a couple of the figures that we'll look at. But I'm talking both about absolute geographic distance, miles, between source site, wherever that source population is from, not the increase field necessarily, but the actual site of origin and the restoration site. In our case, these were various evaluation sites. Latitudinal distance, how far north and south are we shifting it, again from the source site of origin, and specific aspects of what we could think of as climatic distance. The particular work I'm going to talk about here looks at difference in average temperature over a 30-year rolling average between the site of origin, wherever that was, and the evaluation or restoration site. So that was one climatic variable that we're talking about, and another was precip. And by diversity, as I said, I diverged a bit from my blurb as I had originally presented it to Laura, and I'm going to be focusing on not functional diversity in the plant community, but diversity of the actual seed source at a genetic level as it could be affected by selection, whether intentional or unintentional, inadvertent during plant material development. So the first couple of experiments I'm going to be talking about, I'm going to orient you to the general structure of this experiment first, because there's two different experiments that both started with the same basis of work. So what we did, starting back in 2014, was we collected wild harvested seed from primarily scientific and natural areas in Minnesota, prairie remnants, in, as you can see, several, tech challenged, like I said, that's the button. So in what we considered 4 different regions of what is historically the tallgrass prairie region in Minnesota, which if you picture kind of an L, and I apologize that my hands are shaking, I'm going to blame that not on nerves, but on a little too much coffee. So southeastern Minnesota, which is in the green, southwestern Minnesota in this kind of teal blue, west central here in that kind of purplish color, and then northwestern up here in the red. And we collected other species as well as part of the seed collection arrangement we had with the Nature Conservancy and the state of Minnesota, among others. But the species that we were focused on testing were 2 each, grasses, forbs, and legumes. The grasses were little bluestem and side oats grama. The forbs were anemone cylindrica, tall thimbleweed, and wild bergamot. And the legumes were the white and the prairie purple clover. Prairie clover. And for those 6 species, because not everybody grew at the same sites of origin, we sampled from a total of 20 different sites, such that each species came from at least 3 populations in each of those 4 regions. So we had 12 populations that were as close to geographically equally distributed as we could for each of those 6 species. And that mileage range is just to give you a rough estimate of the latitudinal distance between our southernmost and northernmost sites, and our longitudinal distance between our easternmost and westernmost sites. So what did we do with all that seeds? I have, just as a quick aside, a citations slide at the end. Please, of course, feel free to take photos of that for anybody who wants the supporting literature. We go into gruesome detail about what we did with the seeds after we harvested them. But eventually what we did was grew them out in plugs over the winter of 2014, 2015, and then planted those plugs into what I'm going to refer to as a parental generation at a common garden site here in Rosemount, which is about maybe 40 miles or so south of St. Paul. And the reason for that was because we all know, right, that there's going to be maternal effects for each plant that we harvest from out in the field, just due to whatever vagaries of environment happened to that mom plant that particular year that she was producing that seed. And the idea with planting an intervening generation at this common garden is to mitigate to the best we can the effects of those maternal environments. So what I'm calling again the parental generation then is these plugs, which we grew from the seeds which we harvested originally from these prairies. And we had this particular spatial arrangement to try to minimize seed transfer among populations so that we could be reasonably sure that the seeds that were then produced by this parental generation in this common environment were in fact within population crosses for each of these. The first experiment I'm going to talk about was work that was headed by a graduate student named Naomi Rushing, who measured phenology and fitness of the parental generation in this location. But I will then go on to talk about what we did with the seeds from that parental generation, which we then took and outplanted into what was originally 3, but ended up only being 2 outstate sites, which is what these hollow triangles here are indicating. Twin Valley, which is our northerly site, Lake Bella, which is almost on the Iowa border. We would never do this, but we could, if we wanted, have thrown rocks at Iowa from our field. Of course, we would never do anything like that. We would wave and say hello, very friendly. And then we had a third site outside Morris, Minnesota, which would be right approximately here. But things just, for various reasons, just never established there, so I'm not going to be talking about that site. So we seeded seeds in 2016 and then again in 2017 for some species into those 2, the southern site and the northern site, and we grew them out from 2017 to 2022 and collected data from all of those species at all of those sites and replicated transects for all of those years with a couple of sort of hiccups due to the pandemic. And this is the work then that I'll talk about second. Okay, so that's just to kind of orient you so that you have in mind all of this related information. So Naomi's work, so again, what she did was looked at the parental generation in this common environment. And the first thing that she was looking at was there an effect of latitude of origin on, and yes, she's looking at phenology here and not fitness, but of course we know that the timing of when things set flower, especially relative to whether pollinators are available, is going to affect fitness. And so what we've got here is each of these triangles, these blue triangles, illustrates, and again, my apologies for the shakiness here, the population average date of first flowering for each of the different populations. And let me know if you can't hear me as I'm stepping away from the mic here. So as you can see, if you were to trace vertically upwards from the x-axis, which indicates latitude of origin, of the site of origin, so there's one population here, a second population, a third, and a fourth, and so on and so forth. So we've got 12 different triangles there. And the main point that I want to point out about this is that, first of all, counterintuitively, okay, let me rephrase that. Not counterintuitively, but counterintuitively with regard to fitness, what we're seeing here is that significantly for the thimbleweed, the white prairie clover, and the purple prairie clover, the further north the site of origin was, so the stuff off to the right-hand side of each of the three panels, generally speaking, the later the flowering date was, which is what we would expect, but also suggests that if we were to move southerly populations north, that's actually going to shorten their growing season, which could have a negative effect on fitness, although that's not what Naomi was measuring here. So the northerly origin populations flowered sooner, the thimbleweed by an average of 2 days, the white prairie clover by an average of 4, the purple prairie clover by an average of 7, which of course you can see with the angle of the trend line here, that it's a stronger effect in the purple prairie clover. So that's one thing I want to point out, but the other thing I want to point out is just how crazy variable some of these populations are. I mean, yeah, sure. Let me just pick this one as an example here. This is the white prairie clover from our northernmost collection site, which I believe was Pembina Prairie, which is sort of halfway between Moorhead and Crookston, more or less. But you've got dates that run as early as, what is that, about June 20th and as late as the beginning of August. So really, really wide variations in population response here. And we don't want to lose that, right? When we're moving seeds from populations and into production, we don't want to lose some of that variability. Okay, so the other thing that she looked at here is the average how long the flowering period was. And sorry, I didn't correct that here. But the average of the first flowering period. And what we're finding here is that neither the thimbleweed nor the purple prairie clover had any particular response for length of flowering period. It didn't matter what the latitude of origin was. They all had basically a period of flowering that was about the same length. What we saw here with the white prairie clover was, as you might expect, the further north the population originated from, the shorter its flowering period, which makes sense, right? Because of course frost is going to hit whenever frost is going to hit, and if you're still kind of hanging out waiting for your pollinators to show up, you're out of luck. But again, we're seeing really wide variations across populations, particularly for our southern and kind of mid-state populations here. Okay. And just one final point here, that the actual, how many days are we talking? Approximately 8 days of a shorter flowering period, which again doesn't seem like very much, but when we start to take into account the mismatch, the increasing mismatches between flower phenology and insect or pollinator phenology, that could become a problem. Okay, so what are we looking at here? She also looked at fitness. So again, this is the parental population. These are the seeds that we harvested directly from those wild sites and grew out in this common garden and at Rosemount. And what she's looking at for each of these circles, and hopefully these show up well enough. Hopefully you folks in the back can see we've got individual hollow circles here that represent each of the different populations, and we're only showing thimbleweed and white prairie clover here because she didn't find any kind of significant trend for the purple prairie clover. And what we're seeing is that there's two different kind of contradicting effects of latitude difference and absolute difference. So remember I mentioned what we're talking about with regard to distance, and what we're showing here on the x-axis is actual geographic distance, here it's in kilometers, between the site of origin and the evaluation site at Rosemount. What we're showing on the y-axis is the latitude of the source, and this indicates the latitude of the evaluation site. So sites that are above, up here, came from latitudes further north than our evaluation site, and the sites down here came from latitudes that were further south of our evaluation site. And what we're looking at here in this color bar is the fitness scale, and the blue color indicates more fit, this purple color indicates less fit. And the pattern that we're seeing here is that, first of all, if she used latitude and geographic distance together to estimate fitness, that joint prediction did better than either absolute distance or latitude on its own in terms of predicting what the population fitness would actually be. The other thing that she found was that populations that originated from further north, so again, that's going to be the ones that are above and to the right of the triangle, and this was for both species, had higher fitness than species that came from lower latitudes, which is going to be these guys down here. And that once you allowed for latitude, however, the closer the distance, the higher the fitness. And an example of that would be over here. So how do we know that these are higher fitness? Because the color is closer to the blue than to the dark purple. They're at a latitude that's not terribly dissimilar from the location site. And how do we know that? From the value on the y-axis. And the closer we get in terms of absolute distance to the evaluation site, the higher the fitness was. So what are the main take-homes from this? Again, latitude and distance are both important predictors, but not necessarily in the way that we would think, because commonly we're thinking, okay, climate change, we need to move stuff from more southerly populations to more northerly populations because we expect that the future conditions in the northerly populations are going to be more similar to what's happening in the southerly populations now. But that's not necessarily what we are seeing here. Yes, latitude is important for these two species in this experiment, but in a way opposite than what we might expect. Okay, moving on. So that was the parental generation. And I should back up a quarter step to say that Naomi estimated these over 2 years. The data that I just showed you, the phenology data, was over 2 years. So not as long a term of experiment. What we've got here then is the effects on the fitness. So the seeds that were harvested from that parental generation at Rosemount, as I said, were then outplanted into a southern site, Bella, and a northern site, Twin. And I'm going to give you the Cliff Notes version first because the figure that I'm going to show you is really busy, for which I apologize. But I think giving you the highlights first will maybe be a little more useful. So we found that absolute distance and latitudinal difference and temperature difference between the origin sites and the common garden all had an effect, but how strong that effect was depended on the species, depended on the population of the species, and depended on the evaluation site. So in other words, really context dependent. Generally speaking, contrary to what Naomi had found with the parental generation, the southern origin populations had higher fitness in both the southern site and the northern site. But again, to emphasize, lots and lots of variation among populations. You're seeing the theme here, right? That these populations are really varied internal to each one of them. And our upshot from the work that I'm going to show you here in a minute is that using either absolute distance or climate alone, meaning temperature difference, precipitation difference, again, between the site of origin and the evaluation sites here and here, was not a particularly good predictor of fitness, which suggests that we need to be really careful if we're going to do predictive provenancing pretty much based on move stuff from a southern climate to a northern climate. In some cases, especially if that's the only information we have, which of course is sometimes going to be the case, then okay, that's better than nothing and there are data to support that. But when we have other information, such as information about the actual environmental soil or microbial communities at the sites of origin, we should be working with that material as well. So, okay, my apologies in advance for the busyness of this figure. I'll kind of take us through it one step at a time. Okay, so what we've got here, the vertical columns, this one here, for example, all of these pertain to the effect of absolute distance, again, between the source of origin and the evaluation site. The one here in the middle is latitude, and the one here on the right is temperature difference. The vertical dotted lines, which you can see here, for example, indicates the evaluation site. So the evaluation site is at a distance of 0 from itself, and the further you go, we can see in this case, the lower the fitness. We've got thimbleweed along the top row, white prairie clover in the second row, purple prairie clover in the third row here, and then Monarda here. The other thing I want to point out to you is that these horizontal lines here are ideally the size of fitness that we would have for this population to be stable. In other words, the population is not so unfit that the population level is starting to decline. So that's just a reference line here. I know there's a lot here, so I'm going to focus first on the wild bergamot, the Monarda here. And what we can see is that there's an overall trend for absolute distance here in the bottom left-hand corner, that the further we went from the evaluation site, because the evaluation site is here on the left, the lower the fitness. Similarly, the further in latitude, and I'm sorry, I should say that this is all for the southern site. So the further in latitude that we differed, also lower in fitness, which is not surprising. We're at the southern site, so the furthest source populations are going to be our northern populations. And temperature, so the dotted line here indicates the evaluation site itself. So over here on the left, what we've got are sites that are colder than the evaluation site. So sites that are further away, just in absolute terms, sites that were, or sites of origin that were latitudinally different, and sites of origin that were much colder, all of those had the lowest fitness for wild bergamot in this southern site. And that's just what I'm trying to emphasize here. Now for cylindrica, the thimbleweed up here, we've got a similar trend with regard to latitudinal difference, where the further in latitude you got from the evaluation site, the lower the fitness. And similar pattern to the Monarda with the temperature, whereas the cooler sites had lower fitness. Sites that were as warm or a little warmer than the evaluation site, the fitness was higher. So one big picture here is we're seeing very different patterns with regard to species, right? Because purple prairie clover, it does not seem to care one way or another. It's going to do the same pretty much with regard to all three of these predictors, absolute, latitudinal, and temperature difference. And you can get a sense of the variation. Each one of these colored dots is a population. And you can get a sense of just how wide the fitness variation for each of these populations were. Okay, at the northern site, the thing that I want to point out here, so it's laid out exactly the same. Two things I want to point out here. First of all, the pattern from Monarda switches with regard to absolute distance. For the southern site, the further sites had lower fitness. For the northern site, the closest populations had the lowest fitness, and it was actually the populations from further away that had higher fitness. And the other thing I want to emphasize is that purple prairie clover's response was different at the northern site than the southern site. Where fitness was highest was in conditions that were just a little bit to the south of the evaluation site, and conditions kind of correspondingly, sites that had conditions that were just a bit warmer than conditions at the evaluation site. So the big picture point here is that variation, context dependent both in terms of species, in terms of population, in terms of are you growing them out at a southern restoration site or a northern restoration site, which I realize is stunningly unhelpful with regard to the prospect of producing seed, right? How can we possibly start to do this in an efficient way that's going to make seed more available for projects when each one of them wants its own very special conditions and is going to have its own very special response? Where I'm going with this is that these are the sorts of opportunities where researchers can do a better job working with, producers and practitioners in developing the data that would be helpful in answering the questions that you need. And I'm running a little short on time. Okay, folks, okay if I keep going here for a little bit? Okay, I will try to keep it prompt, I promise. Okay, so how do we sum up this work from this set of experiments that Wild Harvest did, then did a maternal effects generation, then did an F1 generation for 6 years out in these outstate sites? Once we account for latitude, yes, the general trend was more local, tended to have higher fitness. However, we can't just assume that taking latitude into account means move southern populations north. In some cases, yes. In some cases, not necessarily. Populations are also highly variable in terms of how they express their local adaptation. In other words, how much more fit or less fit were they in response to what factors? Now, this comes out of Naomi's work, and I didn't show the figure here. But for example, what drove, in terms of climatic factors, what drove the highest fitness for white prairie clover, it really liked cold temperatures in January. Didn't care about temperatures in the summer, didn't care about variability of precip or growing season precip. It cared about winter lows. The thimbleweed, on the other hand, cared about July highs, and it liked really variable precipitation. So again, if we're trying to make generalizations with regard to what climate factors are and what those mean, the response to that is, the data suggests, going to be really variable from species to species. So in other words, broad environmental statements of similarity or broad statements of climatic similarity is not necessarily going to reliably predict how a population does once we've harvested it, produced it, and have it out in our restoration site. Again, I'm sorry, I realize this is not especially helpful from a production standpoint, except to indicate and underscore, again, as we all know, that researchers need to be doing a better job of coming to you folks, the practitioners and the producers in particular, and saying, what data can we try to generate that might be useful to you? Okay, so just to get briefly to the diversity question, again, as I mentioned, I shifted how I was thinking about this a little bit to emphasize on the diversity of the seed population in question and how is that affected by the plant material development process. Now we know, and I've just got a small selection of data here, that it's not universal, but it's quite common that once we start cultivating varieties of native plants, so actually working towards that cultivar release, we know that the resulting plants differ in terms of their trait expression. And I'm going to pick on switchgrass in particular. My apologies in advance to any of you who are switchgrass breeders. We can get into an argument about the value of molecular genetics and using that as a guide maybe during the breakout session. I'm going to blame that on my PhD advisor that I was guided into the world of switchgrass. But we know that the cultivars, they're bred for, in the case of switchgrass, for example, to be pasture grasses. And what do you want? Certain things in pasture grasses. So the traits differ, and we have good data to suggest that the overall genetic variation in those cultivated and varieties is lower. We also know that some traits that can be inadvertently selected for during the seed and the plant material production process, in some cases can be correlated with invasive risk. So for example, you end up with something that's just very aggressive, very dominant, has high vigor. Yeah, that's great in some ways, but it also is going to depend on the recipient community and how that plant behaves, so to speak, in that community. So what I had done previous to the work that I was just talking about was a 3-year field experiment in which I took 6 cultivars and 6 what I was calling commercially produced ecotypes. So minimal adjustment. In fact, the Iowa ecotype was one of them. Minimal in the ecotypes attempts to reduce the genetic variation. All of the attempts were made to keep those as broad a genetic base as possible. The 6 cultivars, for those of you who are familiar with this, included things like Summer, Forestburg, Caven Rock, and several others. And I grew these out over the course of 3 years at different sites in Minnesota. And what I was interested in was basically are we setting ourselves up by creating cultivars of native species for future headaches, is kind of the short version of the question I was asking. And what I found, first of all, was that what we're looking at here is the commercial ecotypes here in the lighter gray, the cultivars in the darker gray, years that they were out in the field, and what I'm looking at here is stand density, basically individual plants per square meter, which I know the agronomists in the room laugh at the idea of ecologists measuring 1 meter at a time, but that's what I had access to in terms of equipment. And what you can see here is that the cultivars start out with a bang, but self-thin by year 2. And by year 2 and 3, there was not a statistically significant difference in the stand density. Yeah, the mean, the average value of the cultivars, they had a few more individuals, but it was not huge, especially when you sort of scale this up to restoration size. I then looked at biomass, and this is just showing the 3 sites. Again, the lighter gray is the ecotypes, the darker gray is the cultivars, and this is looking at above ground biomass. These units, again, grams per square meter. And yes, statistically significant difference here, but again, in terms of actual yield at the restoration scale, we're not talking a big difference here. So what does this tell us? That particularly if what we're concerned about is getting, in this case, switchgrass established in the face of pretty dense competition from weeds, maybe a cultivar is not a bad way to go. But if you don't have that pressure, then retaining the genetic diversity that we're pretty sure exists in the ecotypes is perhaps a better alternative, and having access to that particular range of genetic variation. Okay, also as part of this experiment, what I did was did little mini restorations, 1 meter scale, little mini restorations, into which I then planted at the standard seeding rate that's recommended for switchgrass. It's a co-dominant, but it is not strongly dominant in these systems. So I've got a moderately diverse prairie community into which I had seeded either, cultivars which are shown by the solid circles or ecotypes which are shown by the open triangles. And then what I did was went through and individually censused all of those replicate plots, counting the number of plants of each species, so a full census. And that's the data that we're looking at here. So for example, in this lower right-hand corner, these are all of the communities that were at the site that's called Waseca. This one is at the site that's called Morris. This one is at St. Paul. And the big picture point here is, it didn't matter which kind of switchgrass was planted in, cultivar or ecotype. The communities, regardless, were very, very similar, which suggests that switchgrass is in fact not coming in. The cultivars are not coming in and dominating everybody else. This is essentially the same data, but we're looking at it from an above ground biomass standpoint. So whether you're looking at actual diversity or whether you're looking at something that's more related to ecosystem function, we're seeing some differences in terms of site. Not a surprise, right, to any of you who've done restoration work, but the communities are not separating based on which type of switchgrass is in there. Okay, so what I want to wind up with here, and I appreciate your patience, I'm sorry for running a little bit over, is first of all, with regard to this diversity question, whether you use cultivars or ecotypes is going to depend on the risks and benefits of your particular context, invasiveness, need to establish versus risk of over-invasiveness, for example. Insofar as ecotypes have greater quantitative genetic diversity, so diversity in traits that are important in restorations, pathogen resistance, herbivore resistance, drought resistance, so on and so forth, insofar as ecotypes have greater genetic diversity, they're going to have a stronger capacity to adapt to stressors. Whoops, I'm sorry. So there's an argument to be made for using the ecotypes unless you have a specific need for the cultivar in your project for whatever reason. And what I came away from all of this was that a major gap in the literature, and this hopefully is where perhaps researchers could step in to be useful, is to look at different provenancing strategies, whether we're talking regional admixtures, whether we're talking composite provenancing, and actually building these in as replicated experiments within existing restorations, and looking at, over the long term, how does the focal species that we're seeding in and the restoration community, do those persist better with some provenancing strategies than others? Because I'm not seeing, at least at this point, and anybody who knows better, especially Justin, correct me if I'm wrong on this, I'm not aware of any long-term studies that have been published at this point that have done in situ comparisons of different provenancing strategies within the same site or set of sites. There is one short-term experiment that I list. It's the first list on the citations page that I'll show in just a second. But it's only, I think it was a 2-year study. And can provenancing strategies help enhance community resilience? Okay, thank you for your time. Questions are probably best left during the break, I'm guessing. But I appreciate it, and if you have any questions, I'd be happy to chat further.